Hi all, thank you for tuning into my channel. This is 6B of the Ozone Technology course. In this session, I will be dealing mainly with the continuation of nucleoproteins, and I will be dealing with the ORP, Oxygen Reduction Potential, and some parameters used in application of ozone technology. In this slide, I will be dealing with the nucleic acids that form the basic components of the DNA, RNA, which determine the characteristic of any cell and correspondingly to the human or animal. Now, there are 4 nucleic acids that are the basic components. They form the nucleotide, which is a short chain, and a nucleoprotein, that's a long chain of nucleotides, and finally the DNA chains. The 4 nucleic acids are called adenine, cytosine, guanine, and thymine. Each has got its own letters, got A, C, G, and T. Any combination of 4 of these acids will form a code in the gene of the chromosome. Among the nucleoproteins, the simplest ones are the RNAs. RNA means ribonucleic acids. Ribonucleic acid contain a ribose sugar. This RNA composes of several nucleotides, and their role is to create new cells or to translate the DNA into proteins, acting as a messenger between DNA and ribosomes, and help ribosomes choose the correct amino acids for the template, which is then used to create new proteins of the chromosome chain. There are 3 types of RNAs. The one is the transfer RNA, you call it tRNAs. Transfers the amino acids to the growing peptide chain. They are also called this as T cells. The next one is a rRNA, which is a ribosomal RNA, and this is a catalytic component of ribosomes. That is, the ribosomal RNA can combine the protein to form the ribosome, combines with the proteins to form ribosomes, and finally the messenger RNA. Messenger RNA are ribosomes that combine with the messenger RNA to carry out protein synthesis. It carries the protein sequence and form called template for a specific protein synthesis. DNA is a deoxyribonucleic acid, which is actually the RNA combining, 2 RNA combining with the use of the hydroxyl group, and therefore you have one oxy going, deoxy, you call it, removal of the hydrogen. It is a twisted double helix link of 2 RNA chains, which is a mirror copy, and this comprises the chromosome and the genetic code, which is a character of the cell or the animal. Multiple units of each of any of these 4 of the nucleic acids will form one code. I know it is quite difficult to understand actually the meanings of all these terms which I am referring to. So I've got another diagram that gives you exactly what the mRNA is. If you notice that mRNA, which is the one that carries the information of the protein or the code or the gene. Now, as you know that each protein has got a specific structure of amino acids, and we have in our body many such different type of proteins. They all have to be synthesized, and the body knows exactly what to do. So what they do is, what the body does is, it makes a copy of the protein chain in a RNA, messenger RNA, using the nucleotides, which are formed precursors. That is, the amino acids are combined in a particular order and form nucleotides, and three of them join up together to form a codon. The mRNA is then exported from the nucleus to the cytoplasm, where it is bound to ribosomes, and then together they make up the template for the copying of the protein, whatever protein that the body wants to make. If you see the diagram on the right, you see that code. You have GCU, ACG, GAC, etc. That's a codon which forms a ribonucleic acid. In this, you find there are only three, but with respect to the genes, it will be 4. As I did explain to you, the ribosomal RNA are actually the catalytic component of the ribosomes. Now, this is a picture where I have given you a typical example of a ribosomal RNA. It is a bit more complex than the messenger RNA. It contains 4 different RNA molecules, as I did mention to you. If you notice, it's they call it 18S, 5.8S, 28S, etc. The nucleolus synthesize 3 of them, and the other 4th one is synthesized elsewhere in the cell. In the cytoplasm, the ribosome RNA and the protein combine to form the nucleoprotein, which is called the ribosome. Now, the ribosome itself is more what you call a part of your protein. So the messenger RNA takes a ribosome and combines it and synthesizes the protein, the specific protein with the specific link of the specific acids. You can find very many ribosomes that can be attached to a single mRNA, because the ribosomes are actually carriers of the mRNA. Nearly all the RNA found in the typical cell, that is a human cell, is RNA. The final RNAs are the transfer RNAs. This is a small RNA with about 80 nucleotides. It transfers the specific amino acids to the growing polypeptide chain at the ribosomal site of protein synthesis. During the translation, it has sites for amino acids attachment and an anticodon region. This region is where the recognition is of each particular protein that is being synthesized, and it makes a specific template which has got the sequence using the RNA. Please note that the study of the nucleic acids, DNAs, RNAs are pretty complex, and it is not really necessary to know in detail how they function. I only did explain to you some aspects of it, so when we deal with the ozone reaction with, for example, virus particles, we will be able to understand how we are able to destroy the characteristic of that virus. Now let us go to the next topic, which is a more important topic that is related to application in ozone. This is called the ORP, or Oxygen Redox Potential, or electropotential. This is actually, ORP is actually a tendency of a chemical to be oxidized by the oxidizing chemical. So in a particular reaction, a component that is being oxidized will also have the other component which is providing the oxygen, which is getting reduced. Oxidation is typically where there is an electron transfer out of one chemical to the other, and reduction is when the electron transfer from the chemical is coming to that chemical. The degree and the strength of the transfer is measured in volts. The higher the voltage, the higher the oxidation capacity. I have simplified a little bit on the right with a diagram for you to understand that, for example, let us say A is a particular product that you want to oxidize, and you have an oxidant to oxidize A. So when you oxidize A with an oxidant 1, obviously the result will be A oxide, that is the oxide of A, and then you have a side product which is whatever that you get after the oxidant has given away its oxygen. The time to complete this reaction, let us say, is 1 millisecond. So this oxidant to oxidize A takes 1 millisecond for 1 unit. Now let us take an oxidant 2, which also reacts with A to form A oxide plus side product 2, and the time to complete this time is 100 microseconds. This is about 1/10 of the time taken for the oxidant 1 to react. Now let's take oxidant 3, for example. So if it reacts with A and to form is A oxide, and the time to complete is only 1 microsecond, this means the oxidative power of oxidant 1 is lowest and that of oxidant 3 the highest. So if you translate it into ORPs, you find that, or electric potentials, you find that electric potentials of oxidant 3 is higher than electric potential of the oxidant 1. So we have a table of some of the best oxidants, and from there we can find out what is the potential, oxidation potential of each of those oxidants. You'll note that fluoride is on the top of the table, which has got a potential of 3.06 volts, followed by hydroxide radical 2.80, Then atomic oxygen, which is the oxygen radical, 2.42. Ozone comes next as 2.07. Hydrogen peroxide 1.78. Permanganate 1.68. If you notice, chlorine dioxide comes only at 1.57, and the common use we use in our pools are hypochlorous acid, which is 1.49. Chlorine on its own has got only 1.36, and oxygen, finally the last of the oxidant, is only 1.23. Now, it's very nice to use fluoride because it's the fastest oxidizing agent, but unfortunately it is so fast and so strong that we cannot have actually control it, and therefore it is not possible to use it for the practical terms. So the next one is a hydroxyl radical. Yes, we can use hydroxyl radical and atomic oxygen, both of them, but this only is an intermediary in a reaction. That means you don't have it separately. So you have to have it as part of a reaction, and it's extremely fast, and also we use it in advanced oxygen technologies. Therefore, the most convenient and the best substance we can use is ozone. So ozone is much better than all the others: hydrogen peroxide, permanganate, chlorine dioxide, hypochlorous acid, and chlorine and oxygen. Because of this, we prefer, if possible, to use ozone. We have to measure the redox of any substance that we are oxidizing. So that will tell us how much oxidation has taken place. And for this, we need an equipment or measuring device. This measuring device is a ozone probe. The ozone probe contains two small little probes connected the outside with a solution to be measured and connected the inside by a salt bridge. The potential difference between the two junctions, when connected to the voltmeter, will read the ORP in millivolts. I don't want to explain too much on how the ozone probe works because it is not relevant to our subject. What we have to understand is when you mix any oxidant, ozone or chlorine, for example, or bleach, with the water, the ORP rises, and depending on how much of ozone or chlorine that you use, it can go above 750 millivolts. The theoretical or maximum value you can reach is about 2.07 volts or 2700 millivolts. This has never been achieved in a practical sense. When in our applications, when the ORP goes higher than 750 millivolts, the water is devoid of bacteria, and therefore it can be termed as sterile. Let us discuss now the use of chlorination in water treatment, particularly in swimming pools. Now, you would have heard the term free chlorine, combined chlorine, and total chlorine. Well, the free chlorine and the combined chlorine will become the total chlorine. What happens is when sodium hypochlorite, that is your bleach, is dissolved in water, it will dissociate to form sodium and oxygen chloride, OCl, or hypochlorites. Now, in alkaline solution, which is above 7.8, it remains as OCl. But when you add acid to it, like say for example hydrochloric acid, the dissociation becomes the sodium hypochlorite with water and your acid, you have hypochlorous acid, HOCl plus Na plus and OH minus. So that is the most reactive aspect of in this reaction is the hypochlorous acid, HOCl. You find that if you keep on adding the acid, it will be all tilted towards HOCl and sodium and hydroxyl ion. In above 7.8, the tilt is only to OCl, and below 7.2 and below 7, between 7.2 and 7.8, there's a combination of OCl and HOCl. But once you go below 7, then it will be totally HOCl. Unfortunately, we cannot swim in a water which is very acidic because it will start irritating our skin. And therefore, we can only swim in slightly alkaline water, and ideally the pH of the pool must be between 7.2 and 7.8. You will notice that since HOCl is the most active form of chlorine rather than OCl, OCl will not kill bacteria, HOCl will kill bacteria. Therefore, we need more HOCl than OCl. Therefore, we have to come to an optimum pH to get as much as HOCl as possible, and that is pH between 7.2 and 7.8. Therefore, if you have a swimming pool water above the pH of 7.8, your chlorine addition will not have any effect on your killing of your bacteria. Therefore, the pH is very critical for successful chlorination. This graph shows a relationship between free chlorine, the pH, and the ORP. And you know that for our practical purposes in swimming pools, we need to have free chlorine rather than the inactive chlorine, or the OCl, and the ORP preferably between 700 and 770, and the pH between 7.2 and 7.8. I have drawn a rectangle of the area at which the swimming pool characteristics must be. In other words, we have to have at least 1 ppm of chlorine, which will give about 700, between 700 and 750 plus of the ORP, and the pH will be between 7.2 and 7.8, which is ideal for swimming pools. You would have heard the swimming pool man who comes and maintains your pool say that we need stabilizers. Now, stabilizer, it is actually a cyanuric acid. Why do we need stabilizers? What happens is when you add chlorine or hypochlorous acid to the water, chlorine is hypochloric acid is generated. This is extremely unstable, particularly in the presence of sunlight, and it just breaks down so that the chlorine is totally oxidized off by the sunlight. So you will have less hypochlorous acid in the water. For example, if you use, for example, 100 units of hypochlorous acid into the water, within a few hours, 50% of them will be broken up by the sun, and you will have only 50% left over for your reaction or for bacterial action. So what we do now is we can add cyanuric acid. Cyanuric acid is actually a substance that can absorb your active chlorine, as you show in the graph. It can take each molecule of your cyanuric acid can take up to 3 molecules of HOCl and retain it, and this is more stable in sunlight. As soon as the HOCl is depleted in the water, the isocyanuric acid will release the chlorine back to HOCl, so there will be a continuous amount of active chlorine in the water with the use of cyanuric acid. So cyanuric acid actually acts as temporary storage and releases the active chlorine as and when the active chlorine is depleted by use or breakdown by the sunlight. Unfortunately, you have to understand the cyanuric acid is a combination of carbon, nitrogen, and oxygen, or actually it's called a fluorocarbon if Cl is added, and therefore fluorocarbon is not really good for our health. And fluorocarbons are known to be carcinogenic and toxic, and over a long period of time it may be harmful. So it is very important that we take particular attention as to how much cyanuric acid we are adding, and we have to add only a very limited quantity, just enough to act as a stabilizer. This graph is the relationship between the free chlorine absorbed by the cyanuric acid and the amount of cyanuric acid required. You find that about 40 ppm of cyanuric acid will absorb up to 90 to 100% of your free chlorine. So we just have to add about 100 ppm to ensure that whatever chlorine that you are adding will be taken by the stabilizer and released separately. So we follow this particular standard of maintenance in a swimming pool. Thank you very much for having the patience to listen to the session. And this is the end of the session. Please go to the next session, session 7, for fundamentals in biochemistry. If this is your first tuning in, please visit sessions 1, 2, which gives the program details, and 3 to 6a. And I would surely appreciate that if you click the like icon and the subscribe icon, which will ensure that you get notified as soon as the next session is uploaded. Thank you very much and have a very good day.